Assalamu alaikum. So, so it's a pleasure, as I said, being here again. And today I'll be talking. Now I'm presenting as a scientist, not so much as a WPA official. I'd like to talk about psychiatric genetic research and where we are at right now at this moment in time. Psychiatric genetics is actually a field that has been around for quite a while. As a matter of fact, it has been around for like 100 years, like a century. And it started early on with family twin, oh, wow, this is, with family twin and adoption studies. They laid the foundation for us knowing that there is a genetic basis, that there's a heritable basis. And people then started doing genetic linkage studies, looking at large families, trying to find the genes or the regions that might harbor the genes on the chromosomes for these disorders. Then people also did candidate gene studies, that is, looking at some genes like serotonin-related genes, dopamine-related genes, and so forth, trying to compare cases with controls to see whether there's a difference. Well, this was when I started, like at the end of the 20th century, basically. In 1997, that's when I started. People told me that, well, we'll just need a few hundred families and do a few case control studies, and we should soon have the genes. Well, it didn't work out that way. Then people realized we need to have larger samples, we need to study more genes, ideally the whole genome. This became feasible when the Human Genome Project came to a first conclusion. And around the year 2000, it started when we had the whole genome available, and we could perform, like many other fields of medicine, we could perform so-called genome-wide association studies, scanning the whole genome. And that has been done now for for thousands of traits, disorders in medicine. And now we also do sequencing. That means trying to find new variants that we may not know yet in specific populations, in specific families, and so forth. So as you can see, this is a journey. This is a journey that has been going on for like a century. And again, the year 2000 kind of marked a major turning point because it gave us this technology to move from genetics, that is studying one marker at a time, to genomics, studying the whole genome. We now have big, powerful tools at our hands. We are studying big data. We perform case control studies in thousands and tens of thousands of samples, which is known as genome-wide association studies. When we do that, a regular significance threshold of 0.05, the famous p-value, or the 5% type 1 error that you want to accept, is not possible anymore. We need more stringent criteria because we study thousands of markers. So the threshold that we want to achieve is very small. It's a p-value of less than 5 times 10 to the minus 8. But we can achieve that because we have the power of consortia, meaning we have large samples available. So we run this in large samples, in large consortia, like the Psychiatric Genomics Consortium. And just to give you an idea where we stand, this is just a summary slide of all of findings for schizophrenia, bipolar, and depression. And it's almost outdated now because there are more studies coming out. So what you see here is a so-called Manhattan plot because it's meant to resemble the skyline of Manhattan with skyscrapers. On the x-axis, you have the chromosome number, and on the y-axis, you have the p-value presented as the negative 10 log of the p-value, and you want to surpass that line, the red line here, which is the level of significance, of genome-wide significance. And all these findings above that line are significant findings. These are all markers. So we have now, for all these disorders combined, several hundred loci. And as a matter of fact, just recently, major depression, we put together everything that's out there. We're talking about hundreds of thousands of samples, and we are now at even several hundred loci for major depression alone. And we also study cross-disorder phenotypes. That means combining schizophrenia and bipolar, for instance, to see what is common, what is shared between these disorders. Now, what you shouldn't do is you shouldn't just pick a marker out of these hundreds of markers and say, oh, I'll pick that one and that's it now. That's a cool gene. Well, it may be a cool gene to you, but it only contributes tiny, tiny fraction of overall risk. So we actually think of the genetic background as a polygenic background. That is, you have to take into account the combined effect of all these markers. And you can use that, you can harness that information using a very elegant approach, which is the polygenic profile score. So let's say you have this consortium, hundreds of thousands of samples and several thousands of markers. So you take the whole information and build one score. You can get one number. And so for each and every person, you can build a score. You can say, this person has a high polygenic score for diabetes, high polygenic score for cardiovascular disease, for any phenotype, or a high score for large height. Any phenotype can be studied in this way now. And then you take that score and go to your specific sample. Let's say you have a sample treatment response in psychotherapy or what have you, and you want to see whether somebody with a high score, high polygenic risk score for schizophrenia has, has a less favorable outcome, for instance. You can do this with one score that captures the whole information, the whole polygenic content. And this is a pretty cool tool that we are now using. We have to find out how clinically useful it is, but that's what we're doing these days. So while we did not get what we hoped for 3 decades ago, that is having a handful of risk genes, which would be an oligogenic model, we now know that that it's polygenic. We have made progress because we know it for sure now. So while it's more difficult maybe now, but we have certainty. So this is the polygenic concept, which can be used, as I said, using the polygenic profile scoring method. We also have, and I'm not going into detail here, we have an involvement of rare genetic variation. So in some families, in some populations, you might find some variants that are much more frequent, not so, they are rare, so they are maybe in 5,000 people you will find it 5 times or so, whereas in controls you will not find it, but it comes with a high genetic risk. And these variants are sometimes useful in diagnostic workups of autism, intellectual disability, maybe also in early onset cases of schizophrenia. So there are tests out there that use that information. So we have the polygenic component, and we also have rare genetic variation, known also as copy number variants or CNV. So we have many new findings that want follow-up, that want to be studied in further downstream analyses. How should we go about it? Well, I have a few suggestions. First of all, I think we should use the polygenic scoring to understand etiology, comorbidities, and diagnostic overlaps. We should go deep and longitudinal in our phenotyping. We should focus on treatment and outcome. We should go beyond static genomics to include multi-omics. That means proteomics, transcriptomics, methylomics, and what have you. And we should shift our focus to be more inclusive in terms of the populations we study. Not only the American, European, and maybe Japanese populations or so, but we should go to all parts of the world to study a variety of ethnicities to really make statements that are representative of the whole population of the world. And we should, I think, embed that with psychiatric genetics with global public mental health, because I think it's an genetics is an epidemiological factor, and it should be considered in future approaches to public mental health. Just an example here, how we can use polygenic scoring. So here, this is together with a group in Switzerland. We looked at schizotypy, so a personality trait, if you will. People think, or we think, we sometimes think that schizotypy may be something like a precursor to schizophrenia. And if that were the case, wouldn't we see a connection between the polygenic score for schizophrenia and schizotypy? So here we looked at polygenic scores for bipolar disorder and schizophrenia. This is for schizophrenia, and this is for bipolar disorder. So what you can see is schizophrenia scores predict schizophrenia in other samples, and to a point bipolar disorder, because there's an overlap. A bipolar score predicts bipolar disorder and schizophrenia because of the overlap, but neither a schizophrenia score nor a bipolar score predicts schizotypy. This is like a flat line here. So maybe this whole notion that this is part of the spectrum doesn't hold. Well, this is just one study, it's our study, but it would be nice to see if others come up with the same result. So one use of polygenic scores to see how maybe some personality traits that we consider part of a spectrum actually do fare when you study them in terms of their genetic liability. More phenotyping, deeper phenotyping. So here, using the large sample of the Psychiatric Genomics Consortium, we studied polarity of onset, whether it starts with depression or mania, and age at onset, so when it starts. So we thought it would be easy because this is a sample. At that point in time, it was 60,000 from all over the world. We thought it was easy. We just collect the information, ask the PIs to give us this information. We thought it was easy, but it was not. It took us 5 years to get from the 60,000 samples roughly 15,000 with information on age at onset, and only 7,000 roughly for polarity of onset. So these are actually, I would say, clinical variables that every psychiatrist knows, of your patient. So when you see a patient for some time, you know when it started, you know how it started, like the bipolar disorder with depression or mania. But it's not available in these databases that we have in the consortia, and I think that is a problem. The information is very, very heterogeneous to start with. So when you do this, on the left side, you see the distribution of the age at onset across the various centers from all over the world. So in some parts of the world, it's more an early age at onset. In other parts, it's a later age at onset that might have something to do with the fact that in some countries you diagnose it earlier, and maybe there's some pressure. You could say in America, but there was at one time a pressure by industry to diagnose bipolar disorder more frequently in kids. So that's one aspect. On the right side, you see also the heterogeneity of the definition of age at onset. What is age at onset? The time when you first come to a doctor because you have problems, the time when you are admitted to a hospital for the first time you get an interview, it confirms the diagnosis. So there's a lot of heterogeneity when you do these studies that you have to account for. And we'll do that, we'll account for it, and then we get a result. Here you see we have one significant finding, genome-wide significant finding in the FTO gene, which is a gene that has some bearing on metabolic disorders. Well, that's interesting, but that's just one finding and it needs to be followed up. But what it shows is that actually you can find something with a smaller sample, small, maybe 12,000 here. But think about it, what we could do if we had this information on all of these samples and not just the diagnosis. And we didn't find anything for polarity of onset because this was only a sample of 6,700. So this actually calls for more samples. So again, we could not replicate that in the smaller sample. We could find that there were some relationships in the polygenic scoring. We could see there were some connections or relationships between a high score for autism spectrum, major depression, schizophrenia, also meant an earlier age at onset. in bipolar disorder. And so we can see there is a broad liability, or agent onset of bipolar disorder is influenced by broad liability for psychiatric illness. But I'm repeating myself, we need larger and better characterized samples. To do this, that is in Germany, I ran a study that ran between 2012 and 2019. We collected at various sites in northern Germany and southern Germany, and also a site in Austria. We collected samples in the longitudinal fashion, so seeing patients at least 4 time points, 0, 6, 12, and 18 months. We did comprehensive biobanking, RNA, DNA, serum, plasma, phenotyping at each time point to study the outcome and the course of the illness, because that's an information that never enters these studies. Because there's maybe bipolar disorder and bipolar disorder, but it can be 2 different outcomes, 2 different courses, one with a good outcome, one with a very poor outcome. And this is something that using longitudinal designs we may be able to study much more in much more depth. So this is now known as the PsyCore study. You can look that up here under this website. There's also open for open science. You can reach out to us and work with us on this big sample, because there's, as I said, there's so much data, 2,000 variables, DNA, RNA. We now have basically also proteomics, lipidomics for it. So we have a lot of data that is a treasure chest for analytics. What you can do with it? You can start coming up with new definitions of phenotypes. Instead of saying schizophrenia, depression, bipolar disorder, why not, what we did here, combine this and come up with new patterns, longitudinal patterns. So these are clusters, A, B, C, D, E. These are clusters that may contain like 80% schizophrenia patients, 20% depression patients, and 10% bipolar. They have now there's a new characteristic. Instead of just looking at DSM or ICD, they are defined by a certain longitudinal pattern. And in the future, you might run an analysis on cluster A versus cluster B, and what have you. So these are creative, innovative approaches that I think we should be doing more often in that kind of science and that kind of research. Another important phenotype is response to medication or response to treatment in general. 2008, I started this consortium together with colleagues at the NIH to study the genetics of lithium response. So the genetics of lithium response in bipolar disorder. It's called Condigen. It's a worldwide endeavor. We came up with a retrospective lifetime phenotype assessment to define what lithium response means. And this created the largest consortium ever to study that, more than 2,500 individuals. It was studied in a genome-wide association study. And as you can see here, we had genome-wide findings in this chromosome 21, and in a sample of only 2,500. But it tells you that if your phenotype is quite homogeneous, like lithium response, you might pull out a lot of information that otherwise you wouldn't get. What these genes do, we don't really know. I mean, it's actually more of regulatory nature. These are long non-coding RNAs. But again, this is not the message here. The message is that by defining a well-described, circumscribed phenotype, you might be able to have more power to pull out results, to get results in the future. So that's what we're working on. We want to get this project going on a larger scale. We published that back then in The Lancet, and it has inspired a lot of people. And now we have trained people all over the world, in Latin America, in Ethiopia, in Khartoum. And this was one of the best meetings we had. It was all these people were highly interested, and as you all know, it's a disaster what's happening south of the Egyptian border here. So I hope that one day we can continue these things. Again, all over the world, we are doing these things. Same tragedy here in Kiev. We haven't been able to do that because of the war happening right now. But again, I always encourage everyone to reach out to me and to join if you're interested in genetics of lithium response. So we have many sites. The red ones are the sites that have already participated. The yellow ones are the ones that have expressed interest, have had a training, or have already started. And we are now looking into a second phase of recruitment. So it's a large consortium, worldwide consortium that you are welcome to join. This is pharmacogenetics, right? Studying the genetics of response to a medication. You may be familiar with that concept because there are already tests available out there that tell you which medication, or that promise to tell you and the patient which medication to choose to predict response, to predict side effects. However, I would caution against an overly optimistic use. There are many companies that produce these tests, and everybody can do that. If you have some money, you can come up with a test and sell it. At a conference, have a company and sell it and make a lot of promises. But you know, many of these tests are not based on a lot of hardcore science, not based on large samples. So we have started a European Union consortium project to do exactly that. And we're just focusing on the classic CYP450 enzymes, the genetic variants of these enzymes, to see whether a treatment that is based on the genetic information is better than a treatment not based on the genetic information. And this is a consortium that we have all over the world now, several sites, and we have started recruitment. It will take a few years until we reach the desired number of patients. And I also would like to emphasize, and this is now my WPA hat here, that it's very important to have meetings like this, to have meetings of the WPA that teach, that educate people in this area about this topic, because there's a lot of novel technology out there, there's a lot of promises made, and we need to judge this, or we need to appraise it openly and in an honest fashion. And we'll do this here at the WPA meeting that we had in Kolkata in India last year. We did it in Abu Dhabi last year. We did it in Yerevan in Armenia. So where we engage the stakeholders, people like you, clinicians, researchers. We also talk with patients about the prospects of genetics, especially pharmacogenetics. So it's really something where WPA can make a difference, and I hope will be making in the years to come. I also mentioned that we should not only focus on genetics, or not even only on genomics, but we should go multi-omic. That means look at the products, look at the proteomes, the proteins, the proteome that comes out. Look at at what happens at the transcription between the genes and the proteomes. Look at maybe the epigenome, the epigenetics. So there's so many omes that you can study. We have been studying this in proteomics. We have come up with various patterns here for schizophrenia and bipolar disorder, so you can see differences. We have studied lipidomics. That was the largest study ever to be done, or the first to study lipidomics. And we're not talking about cholesterol. We are talking about all the lipids that are out there, lipids that you've never heard of, lipids that play a role in cell adhesion, in cell-cell contacts, lipids that are, again, fancy names, but lipids do play a major role in the functioning of our organism. And so we did this in schizophrenia, bipolar, and depression. We did it in a trans-ethnic sample from Germany, Austria, Russia, and China. And we came up with some interesting results differentiating cases from controls and so forth. So this is, again, I think, this is published in JAMA Psychiatry last year. And finally, I would like to emphasize that I don't consider genetics, genomics, multi-omics, something that should be done in the ivory tower. It is something that all of us, I think, at one point, we should look into that. We should carefully appraise the findings, and we should be ready to embrace these findings one day. And I truly believe that one day we'll incorporate these findings, not in a way that we say, this is the gene. No, as I said, you will study a blood sample for polygenic content. You will have imaging data, you will have proteomic data, you will have transcriptomic data, and these will be variables as valuable as information that you get from a family, an interview with a family member. So I think there will be a whole array of data that we'll be using. And again, it shouldn't only be done in the so-called global north, but also we are doing studies on trauma in Ethiopia here, where at one point this also will help, because there are not so many resources in many parts of the global south. So having information on pharmacogenetic information, having genetic information on response might help reduce costs. So it's not only something for the global north, it's for everyone, for the whole world. We also do study here in the Kurdistan region on the Yazidi population, but also on the population that was terrorized by Saddam Hussein back then. So with the gas attacks, so we look at trauma. And of course, our world is full of trauma, as we know, and it's our duty to study that, well, no, to treat it, to prevent it, but also then to study mechanisms so as to be able to do something from a medical point of view. Well, so I would like to thank all of you for your attention. I would like to thank again the organizers for inviting me. It's the second time I'm here, and as I said before, it's always a pleasure